Thank you so much. It's a great pleasure to be here. And so what I'm going to do today is talk to you about the taste system, and I'm using the word here, taste, really to mean flavor. So as we go through this, let's see, everyone's having problems with this, so I'll see if I can get it. You have to like really push it hard. There we go. You know, if we think about our dietary choices, they are influenced by a whole host of factors. And as we make choices, they influence our internal state, our hunger, our satiety, our body weight, and so forth. Our perception of these foods, of these flavors, also is influenced by our internal state, and in turn drives, influences a variety of external factors that further affect our dietary choices. So this is really a cycle, and if you come in any place in the cycle, you can talk about the arrows going in either directions. What I'm going to focus on today really is how our internal states influence our perception. And when we think about flavor perception, it does include visual stimuli, it includes texture, it includes auditory stimulation, but I'm going to be focusing today on taste and smell. And the internal states that I'll be referring to, first, the metabolic states, as typified here by fasting or satiety. And we can see that in these two different states, there's a number of different circulating factors that sort of typify these conditions. And these factors circulate in your bloodstream, they're present in saliva. And the taste and olfactory systems are exposed to these factors as they shift in response to different internal states. I'll also be talking about the immune system a bit. And I kind of led myself into this because as I started thinking more about obesity, diabetes, metabolic syndrome, and so forth, you almost can't separate these internal states. They're very tightly integrated, and when you're looking at conditions that alter things like leptin and so forth, many of these conditions also influence your immune status and vice versa. And there's some really exciting work going on in both of these areas in the taste and smell field. So I'll be touching on both of these areas. So if we think about how can internal states affect your sense of taste or smell, one of the ways is that it could actually alter the structure of these systems. As an example, the number of taste papilla could be affected. So if we have, we do know that people with lots of taste papilla tend to be more sensitive to tastes across the board. It can also affect the amount of a particular receptor protein that's expressed. And we can study both of these this is a human tongue, not mine, one of our brave volunteers. And one of the studies, as an example, was studies that were done with subjects. We know that there's a specific gene for specific bitter taste that varies tremendously in the population. Some people have a version of this gene that works really well. You're really sensitive to bitter compounds, and some people can't detect them at all, these certain bitter compounds. But there's also a big chunk of people in the middle, and there's intermediate forms of this receptor that are moderately sensitive. But it turns out within that middle population, there's also, people that are more sensitive or less sensitive. And the reason for this turns out to be the amount of receptor protein that's expressed. So these people all have the same genotype of this particular T2R38 receptor protein, but these people over here perceive the same bitter stimulus much more intensely than these people over here. And that's because of the amount of receptor protein that they're expressing. So what are the factors that might affect that? And so that's what kind of has led a lot of the researchers to start investigating this type of question. Another way that our internal state can influence the sensitivity of our taste and smell systems is to modulate the input, modulate the actual function of these systems. And so in this case, I'm showing now an olfactory cell here. So odors come in, they interact with receptors in the cilia. They bind to these proteins. Here is an olfactory neuron. Here's a couple of taste cells as an example. This binding triggers a whole transduction cascade within the cell, and that leads to activation of electrical currents. In the case of the olfactory neuron, it is a neuron, so this is the nerve fiber. In the case of the taste cell, it releases ATP from the taste cells, or a neurotransmitter which acts on the sensory afferent fibers to send the signals to the brain. So you might have a situation where under some circumstances the output looks something like this, but perhaps under another set of circumstances it looks like this. So you have a much enhanced response to the same stimulus. And I'll be talking about, or it could be suppressed, and I'll be talking about some examples. So I'm going to start with olfaction, partly because there's not been as much work in olfaction in this area, but what has been done is really interesting. So we know that the taste of food relies both on taste and smell. And in smell, we get odors through the front of through our nose, orthonasally, as well as through our mouth. And that's called the retronasal pathway. And so as we're chewing, odors are coming up through the back of the mouth into the nose. And the odors that come through this pathway actually interact with particular subsets of olfactory receptors that are connected to the olfactory bulb and actually activate a part of the brain that is directly integrated with the taste information that's coming from the mouth. And so that's very hard to discriminate this type of retronasal stimulation from taste. So that's, we call food doesn't taste right or it tastes good, but really it's mostly smell. And that's why it's so hard for us to discriminate these. When we looked at the olfactory pathway, we discovered that there's a lot of hormonal factors expressed in the olfactory system. And this is just a table from a recent review paper from Debbie Fadul's group. And if you look at, in the peripheral olfactory system, you can find ghrelin, you can find insulin, leptin, cholecystokinin, and so forth. And in the central components, in the olfactory bulb, you see a whole host of these factors and receptors for these factors within the olfactory bulb. So it's a very rich and sensitive to the metabolic environment. But how does this actually work? And so I'm going to kind of summarize a lot of work in the interest of time. But using a lot of electrophysiological methods in mouse models and so forth, this is sort of getting to the mechanism of how this might work. We know that insulin actually can modulate the activity of the olfactory neurons that detect the odors. So these cells actually express insulin receptors, and the insulin triggers a pathway that modulates the potassium channel in these cells to alter its sensitivity to incoming information. And so insulin and glucose levels in the brain actually influence the sensitivity of these olfactory neuron pathways. And looking at the other extreme, going to the bigger outcome measure, it has been observed that morbidly obese subjects exhibit reduced olfactory sensitivity and are more likely to have absolute olfactory dysfunction or anosmia. So that's sort of two extremes from the zoomed in to the molecular mechanism over to the large systemic effect. But what's been found from a variety of studies in mouse and now in humans is that generally speaking, conditions where the the hormonal milieu is suggestive of a fasting condition. You see an elevation in the sensitivity of the olfactory system. And in contrast, conditions where you have a hormonal environment that is similar to satiety or a fed state, you see generally a downregulation of the sensitivity of the system. So the system is sort of tuned. When you're hungry, you're actually more sensitive to odors. So insulin modulates the sensitivity of the nerve cells in both peripheral and central parts of the pathway. It also functions in the ongoing regeneration of the olfactory epithelium. And so this is more of a chronic long-term effect, but we also see that diabetic rats, for instance, recover more slowly from damage to the olfactory epithelium. So if you damage the olfactory epithelium with a chemical stress, eventually it will recover, but in the diabetic condition, that process is impaired. We don't know, however, whether these metabolic effects act selectively to shift sensitivity to certain types of odors. You might imagine that hunger should make you more sensitive to food-related odors, but maybe not to non-food-related odors. There's been a little bit of work in that, but it's pretty equivocal. It's not really clear that it's that selective of an effect. Okay, so moving on to taste. In addition to other aspects of flavor, we have a variety. So taste really is bitter, sour, salty, sweet, or umami. These are the main components of taste. And when we think about different taste qualities, bitter really is the primary quality that drives food rejection, and sweet is the primary quality that drives appetitive behavior or attraction. How many people had dessert after lunch? All right. Most of the work that's been done in this field, and I'll point out a couple of other areas, but most of what I'll be talking about has to do with looking at how hormonal and immune factors influence sweet and bitter taste. So a few taste basics. Taste cells are replaced throughout life. So both taste and olfactory epithelium are regenerative tissues, and these cells are replaced about every 10 days or so from a population of stem cells in the taste bud. So within each taste bud, you have basal cells, you have differentiating cells, and you have a variety of mature cells, as well as some degenerating cells. And we have different markers for being able to look at these different cell types. And we also have different types of taste sensing cells. So we have what we call type 1, 2, and 3 cells. The type 1 cells are thought to be predominantly responsible for salty taste. Type 2 express receptors for either sweet, umami, or bitter compounds. And type 3 cells are thought to be primarily sour sensing cells. And we have a lot of different proteins that we know characterize these different cell populations. So moving in a little bit farther, there's two major taste detector types. Ion channels, which account for predominantly salty and sour tastes, and receptors. These are G protein coupled receptors. There's a couple of different pathways for glutamate perception. Sweet detection, this is showing the T1R2, T1R3 heterodimer. There's a second pathway for sweet involving glucose transporters that I won't be talking about today, but is an important component of this sweet perception. And about 25 different T2Rs that detect different types of bitter compounds. And we know that there's a lot of genetic variability, as I mentioned earlier, in these receptors. There's versions of sweet receptors that are more sensitive to sweet and others that are less sensitive. And likewise, as I mentioned earlier, with the bitter. But the genetic variations only account for a small proportion of the observed variability in our taste sensitivity and our preference. And so the question becomes, what's accounting for the rest, and how does that work? So if we think about some of the systemic shifts, and these, again, I'm starting with thinking about the big outcomes where we're really looking more at associations than mechanisms. It's been reported that type 2 diabetics exhibit selectively reduced sensitivity to sweet taste. In diet-induced obesity, in animal models, show reduced sensitivity to sweet and umami without a difference in bitter perception. And in DB mice, they lack a functional OB receptor. They have an increased neural response to sweet. They may have hyperglycemia, but they actually are in a state of where the cells think they're hungry, and a higher behavioral preference for sweet compounds. So these are very global manipulations or alterations, and there's a lot of different hormonal changes going on, obviously. Under conditions of low energy, what we see is in short-term fasting, the humans, these are both human studies, more sensitivity to sucrose and salt without a difference in quinine perception. However, in a shorter-term study that was just looking pre versus post-meal, it was reported that there was no difference in taste thresholds. So the time course and the specific shifts in the hormonal environment obviously can make a difference there between those. In caloric restriction, these are animal studies. Some anatomy studies have shown that there's actually altered taste cell populations at 30 months in calorically restricted rats versus ad-lib fed rats. And this was a selective effect on specific subpopulations of the taste cells. So there was a reduced number of these. So this gusducin and T1R3 are markers of the type 2 cells that express the receptors for umami and sweet. And also was reported that there was more GLP-1 and leptin positive taste cells, and I'll talk a little bit more about that in a minute. In bariatric surgery, a number of studies have been done now, and we see increased sensitivity to sucrose and fat, particularly at high levels of concentrations. Reduced expression of gusducin in the taste system, again, a marker of taste cells, of mature taste cells, in particular sweet and umami sensing cells. And preference shifts to lower sugar and fat. This is in another human study, a laparoscopic sleeve gastrectomy. Did not, however, see a change in sweet taste thresholds in adolescents. So there may be differences, but again, unless you know more about the specific hormonal milieu, not just in the blood, but also in the saliva and in the taste system, it's hard to draw general conclusions. And the other point that I wanted to make is that these systemic alterations in metabolic state also induce shifts in the microbiome, certainly the gut microbiome, and almost certainly in the oral microbiome, although I think we need, there's not been a lot of work in that area. I think it's important to start thinking about because, as I'll talk a little bit more about when I talk about the immune system issues. So if we look at leptin, taste and obesity, one of the interesting studies showed here that as you look across the day, plasma leptin rises from 8 a.m. to 2200. And in a normal weight subject here, as you look at the diurnal pattern here, and you look at taste thresholds, sucrose taste thresholds also rise. And this association between sucrose taste thresholds and leptin has been seen in a number of studies. But this shift doesn't occur in obese subjects. So there's something different going on. They start out basically similar, but the obese subjects are not. Obviously, their taste system is not responding to the shift in leptin. So if we look at the leptin and ghrelin pathways, generally leptin suppresses appetite, ghrelin stimulates appetite. And the leptin receptors are expressed on sweet-sensing cells, so specifically. Lower salivary leptin is associated with higher sweet sensitivity. And leptin administered in an experimental model suppresses the taste cell sensitivity to sweet stimuli. And in another model, again, similarly, where there's reduced leptin receptor and increased GLP-1 expression in taste cells, also exhibits enhanced sweet preference. It's also been shown that ghrelin is produced in taste cells, and in mice that have knocked out ghrelin receptor, there's a reduced sensitivity to salt and sour. So this is really the only report implicating metabolic control of salt and sour taste. I don't think that that means it doesn't happen or that it only happens rarely. It just hasn't been studied nearly as much. So again, to summarize, a number of research studies done primarily in the Yuzo Nomiya's group in Japan. If we look at leptin and endocannabinoids, the conditions which shift the ratio of leptin to cannabinoids shift sweet sensitivity. So under high leptin and low cannabinoids, we see suppression of the sensitivity of these cells, and in the opposite in low leptin and high cannabinoids, they're more sensitive. And this actually is happening at the level of the receptor cell. And there's been electrophysiological work, again, showing that it's shifting the sensitivity of certain ion channels, resulting in increased neural output. So a hypothesis might be that the ratio of leptin to cannabinoids in taste tissue sort of serves to tune the taste system to drive the behavior needed to restore blood glucose and energy levels. So what about GLP-1? Is another one of these metabolic hormones that people look at all the time and measure and have talked about as potential ways of controlling weight and body composition and so forth, and food intake. So GLP-1 is generally low with fasting. It increases post-meal. It reduces gastric emptying and inhibits food intake. So what does it do to the taste system? Well, as I mentioned, GLP-1 and its receptor are expressed in these type 2 taste cells, and so they are sensitive to this hormone. And in particular, they're primarily expressed in the sweet-sensing cells. If you, in the knockout mice, GLP-1 receptor knockout, they have a selective deficit in sweet sensitivity. And here I'm just showing some data. This is nerve recording data. So this is the response of the taste nerve to sucrose here in the wild type and in the knockout. And sucrose, glucose, maltose, fructose, saccharin, sucralose, you can see are all different, whereas there's really no effect on salt, bitter, monosodium glutamate, and sour. So this seems to be a pretty specific effect. And if you look at the behavior, it is translated in the behavior. And so there's been a couple of studies that were relatively, that were pretty consistent, actually showing that in wild type, this is showing the wild type, so this is the lick ratio versus water. So this is showing their relative preference for the sweet solution or the taste solution versus water, with increasing concentrations of sucralose. You can see the wild-type mice increase their relative intake to the taste relative to water, whereas the knockouts are less sensitive. Similarly with sucrose, interestingly with monosodium glutamate, the effect was the opposite. So they saw a lower relative preference for MSG to water. These are brief access lick tests in the animals. So they had a reduced preference for sweet and an enhanced preference for MSG IMP. So I'm using the term preference here intentionally, as opposed to sensitivity. This particular method is a method for preference. They're showing a preference for the taste stimulus versus water. And if you're reading taste literature, you need to really look at how the data are presented. This is not absolute sensitivity. In an earlier study by Ninomiya, what they looked at MSG using a lick rate. So this is the number of licks in 10 seconds. And they saw that there was no difference in the lick rate before the wild type versus the knockouts. And so that means that the mice can probably detect the material. They just liked it better. Right? So those are two different measures, and they tell you a little bit different. They give you a little bit different information. So to summarize, some of these, a lot of these studies here for the hormonal effects, under conditions that look like satiety, where you have high leptin, low ghrelin, high GLP-1, animals are generally more sensitive to bitter. They're a little bit less sensitive to sweet, and maybe a little less sensitive to umami. In hunger states, or states that look like hunger, we see an increased preference and a sensitivity to sweet, potentially an increased preference for umami, and a similar dislike for bitterness, but it's not enhanced relative to normal. So we might present this as sort of sweet and bitter taste are really kind of being tuned to drive behaviors towards food seeking or food avoiding, and to what types of foods do we need to get. In calorie restriction, one of the interesting findings is that there's actually more GLP-1 and leptin in the taste system. And so these little faces here is sort of neutral. You might expect calorie restriction to look more like hunger, but it actually looks more like satiety with respect to sweet and umami. So this might be that what they've seen is that there's fewer sweet and umami sensing cells in these mice. So maybe this is some type of an adaptive response. So moving on to immune response. There's a lot of, and this is probably the simplest immune response diagram I could find anywhere, because I could fill the whole room with immune response diagrams, right? But if, the components that I'll be referring to, pathogen-derived molecules activate the immune response via toll-like receptors. So LPS, which is lipopolysaccharide, which is a component of bacterial cell walls, acts on these toll-like receptors. We also have TNF-alpha, and so these factors sort of stimulate the pro-inflammatory pathways, and IL-10 is among the factors that kind of damp down the immune response. Again, this is probably the most grotesquely simplified version of the immune system you'll ever hear. But what the initial observation was that LPS stimulates which, using LPS to stimulate an inflammatory response, inhibited taste cell proliferation and shortened taste cell lifespan. So when this was observed, it was really the first recognition that the taste system actually is modified by these potential disease states or altered immune states. And so these Han Wang and others, this is just showing the expression of these toll-like receptors, which is one of the key receptor populations in the immune pathways, are present throughout the taste cells. These are all of a variety of different toll-like receptors. So they're clearly very sensitive to the environment of the immune milieu. So looking at TNF-alpha, TNF-alpha is elevated in high-fat, fed, obese mice, right? So how much can we learn, so how much is hormones, how much is these immune factors? These mice have fewer taste buds and slower taste cell replacement, and knocking out the TNF pathway didn't alter the number of or subtype of the taste cells, but it reduced the sensitivity to bitter stimuli. So this is showing the relative response in the wild type versus the knockout mouse. So they're less sensitive to bitter compounds. I'm sorry, they're more sensitive. Their threshold is lower, so they're more sensitive. And for IL-10, low levels of IL-10 are found in obese subjects with metabolic syndrome. And in IL-10 deficiency, we see fewer taste buds and fewer taste cells per bud. So this IL-10 pathway, also when IL-10 is low, under metabolic states or internal states where IL-10 is low, it may be contributing to the suppression of taste, particularly sweet taste, in conditions such as obesity and elsewhere. So just to kind of summarize, where we have inflammation, we know in a variety of states where we have inflammation that can influence this proliferation, differentiation, cell death pathway. And it seems to affect the taste system rather selectively to make it specifically more sensitive to bitter. So conditions that promote inflammation, obesity, et cetera, alter taste function, we see reduced taste function overall, but enhanced bitter sensitivity relative to sweet and umami. So I'm hypothesizing under conditions of stress that taste function is altered to drive a shift in dietary choice to fulfill a perceived physiological need for more energy and greater protection against potential toxins. If you're already sick, you really want to avoid that potentially toxic plant. So the key points, really, food preference is driven by sensory cues. How we detect and interpret these sensory cues is really shaped by our hormonal and our immune state. Taste cells are influenced by factors in the blood and saliva, but they can also release many of these hormonal and immune factors themselves and may be able to do a fair bit of self-adaptation that we really need to understand better. Under sort of energy replete conditions, there's generally reduced sensitivity to sweet and enhanced sensitivity to bitter. Whereas in inflammation and disease, we see reduced regeneration as well as more sensitivity to bitter. So my challenge is, how can we shift choice when an internal state is fueling a drive for sweet and avoidance of bitter? Can we fill up our taste buds instead of our stomachs? Maybe we need a DRI for taste. Thank you. Questions?
We do have a couple minutes for questions, but I had one and I'm formulating it still as I'm thinking about it. So I guess all these external factors like satiety and being hungry, How about the emotional states that we're in, the stress eaters that we are, or the hangriness, when people get hangry? How do any one of those, affect the taste perception? And as you said, how can we prevent?
So my next study, I'm looking for volunteers to come in. I'll make you really pissed off, and I'll see what your sense of taste is like. So I think as far as emotional state, I think that definitely can play into it. We do know that there's elements of the cholinergic system and the adrenergic system also influence the function of the olfactory pathway. In an alarm state with an animal is presented with a potentially a predator, for instance, or a loud noise, you do see an increase in the sensitivity of their olfactory system, right? They're cued in because they need to make that, they need to be very sensitive to their environment. So I think, that's sort of the one little bit that I can speak to that, but really it hasn't been studied in any organized way. It's a really interesting thought. Yes?
Thank you for a wonderful, wonderful talk.
Thank you.
I was wondering, in chronic mental illness, where people are on a lot of different drugs, almost all of those drugs are orexigenic, and I wondered if anybody's looked at that. Is it a change? Do they have changes in taste sensitivity? Because they gain like 20 pounds as soon as they get on the drug.
Yeah. There has been some work done on that, and there's actually, I think there was a recent meta-analysis, and I don't know how well it was done. I haven't seen it, but I think the bottom line was that, especially with polypharmacy, there is a suppression of the taste and olfactory systems generally. And certainly, if you look at any drug in the pharmacopoeia, one of the side effects is taste and smell dysfunction. These systems are, as I said, adrenergic, cholinergic. All the anticholinergics suppress olfactory sensitivity. So yeah, it is a big issue.
Nancy, question: would there be a gender difference, especially when you're talking about hormonal influence on taste, so that in the hungry state and in the satiated state, would you see differences in terms of sweet and bitter preference in the male and female?
Male and female. There haven't been a lot of studies. I think the study that Yanina Pepino did in following bariatric surgery, she did see a difference in males versus females. And my recollection is that the change was actually more evident in males than in females, but it wasn't clear enough to say that it was gender because there were other factors that were different between the groups. So we need to look at that. Obviously, the hormonal environment is going to be different. We know there are endocrine, there's oxytocin receptors in the olfactory pathway. There's other, estrogen receptors on peripheral olfactory receptor neurons that we don't really know what they're doing there yet. But it certainly is important to look at. Yeah, one more.
Sort of along that same line, if you changed your diet, would that change how the receptors?
Yeah, great question. So there's been 2 studies, count them, that looked at that. The first one was with salt, and it looked at a long-term reduction in salt intake on salt taste sensitivity and preference. And what they saw was that over a period of a few months, you did see a shift in sensitivity so that the same concentration of salt now was perceived as more intense after that low sodium diet. And that helped to set the guidelines for the IOM guidelines for lowering sodium sort of incrementally, that people do sort of adjust to that. For sugar, however, there's only been one study done so far, and it was sort of a pilot study, a small study, where they reduced sugar in diets of people. for 3 months. And what they saw in that study was that they did see a shift in perception in terms of the same concentration of sweetness in the pudding and the two foods that they were using as test foods in the beginning versus at the end of the diet change. So they did report a higher perception of intensity for the same amount of sugar. However, when they then asked in a different setting what was their preferred product in that set of products, they still preferred the higher level of sugar. So they did see a shift in perception, but at least over that short period of time, it didn't change preference, right? So now we're embarking, actually, Gary Beecham and Paul Wise will be embarking on a large study in collaboration with the USDA to look at that in more carefully, because that early study also, it was small number of subjects and they didn't have as good control over the diets as you would like. But I suspect that it will be harder with sweet than it is with salt.
All right, well, thank you, Dr. Rawson.
Thank you.